Okay, good morning, everybody. Thank you so much for this kind invitation. I'm going to share with you the rational approach to molecular diagnosis in biology in Africa. This is our experience from Egypt, Cairo University, one of the biggest tertiary center in Egypt. First of all, with the ongoing discovery of novel inborn error of immunity gene and improved definition of clinical phenotypes, the collective prevalence of primary immune deficiency is expected to be around 1 every 1,000 to 1 every 5,000, which is much bigger than was previously reported. The association of the rate of the inborn error of immunity diagnosis and the rate of disability adjusted life yearly is very high in the MENA region, reaching about 20,000 every 100,000 of the population, especially in the MENA region, which have got high consanguinity, as you can see. The molecular diagnosis, just a minute. Okay. The molecular diagnosis of pediatric inborn error of immunity influence the management and the decision and alter the clinical outcome through early use of targeted and curative therapy. Patients without a genetic diagnosis usually undergo a lot of diagnostic odyssey, including numerous specialist referral and exhaustive number of expensive and often unhelpful testing. So the question usually is, how much does it cost to perform a genetic diagnosis for these patients? And we have to weigh the cost versus the benefit of doing the genetic testing. These depend on the diagnostic method we are going to use. The Sanger sequencing costs around $200, but it's only available for just small genes and with very specific phenotypes. The next generation sequencing costs around $450, but it's available only for large gene and when patients have overlapping phenotypes. Whole exome sequencing costs around $800, and it is done when the next generation sequencing failed to detect the mutation in the patient. Second, we are asking ourselves about the availability. These include the availability of kits and reagents that is going to do the tests, the availability of the equipment that we are going to do it, and the availability of personnel who are well trained for the know-how to perform the test, to select the patient, and finally to reach the final diagnosis based on having the bioinformatic analysis and proper diagnosis of the patient. Also, the availability of other alternative methods of diagnosis. These depend mainly in Egypt. In our experience in flow cytometry, we have got since 2010 a lot of experience in using the flow for the diagnosis of patients. Now there are available panels like the Euroflow orientation tube for screening of the different phenotypes of PID, and also the availability of wide array of antibodies that are used for many of the intracellular proteins and even for doing the functional assays for the patients. So in 2020-22, we have published our experience from Cairo University on the genetic testing in Egyptian patients with inborn error of immunity. And we describe the achievement in this field in Egypt over the past decade and the optimum way to use the available genetic tools to reach a refined, precise diagnosis. So this is our journey for inborn error of immunity. It began in 2010 by doing just a basic panel for immunophenotyping of the cell, B and T cell markers as well as NK. This allowed us to diagnose patients with severe combined immune deficiency, patients with just CD4 lymphopenia, and those with B cell defects. Later on, and every year, we add more and more of the use of flow cytometry for the diagnosis of patients by adding HLA-DR to diagnose MHC class II deficiency, CD18 and CD11 for diagnosis of lead patients, and then DHR for diagnosis of CGD. Later on, we add the net pH component, so we now can categorize the CGD patient into either P47, P67, or P22, and this confirms the diagnosis and allows us to know which patients are autosomal recessive and which are X-linked. Then we add a lot of intracellular protein like DOC8, WASP, LRBA, proton tyrosine kinase, and ending up in 2022 for the use of Euroflow PID orientation tube panel for screening of all patients we have got in our lab. At the same time of using the flow cytometry, we start collaboration and networking with many other centers worldwide that are interested in receiving samples for diagnosis of the primary immune deficiency. We started to make the best use of the reports we receive from these centers to know what is the specific phenotype of the patient related to the mutation we find, to build up our own knowledge of how to make the best use of this genetic testing. Especially there's a lot of obstacles we have in this collaboration, as most of these projects are, most of these centers are having ongoing projects that have time frame. Only they receive samples in this time frame. Also, there are a lot of regulations coming every now and then for the transportation of DNA samples and couriers to other centers worldwide. And these centers are usually selective in taking patient sample, either specific phenotype, or some centers only take patients who are still living and not dead persons, so they can do functional assays. So our journey, as I said, started in 2010. We started in our center to do the RAG1, RAG2 Sanger sequencing. This is the best fit gene to start with because the patients have a specific phenotype, which is T-, B-, NK+. Also, the genes of RAG1 and RAG2 are very small in size. It's only 2 exons, and each exon has only 1 coding exon. By analyzing this data, we found that 83% of the patients screened for RAG1, RAG2 are for this phenotype are RAG1 and RAG2, and that RAG2 is much more common than RAG1 in Egypt, and we have 2 more mutations that usually present in the patient, glycine 35 valine and arginine 229 glycine are the most common mutations found in this RAG2 gene. In 2014, we started collaboration with Necker. They received sample for the CGD patient and make Sanger sequencing to detect the mutations. We found that patients, by analyzing their data, most CGD P22 patients have only the same mutation, which is deletion for 6 base pairs. So we started this mutation to be done in Egypt, and we diagnosed 10 more patients with the same mutation. And a patient with P47 also had the same mutation, which is a deletion for 1 base pair. In 2015, we started collaboration with Freiburg in Germany for diagnosis of hyper-IgE syndrome, and we started to do Sanger sequencing by collaboration with Erasmus for the T-, B-, NK- skids. The data we received from this collaboration show that DOC8 is very common in Egypt for this hyper-IgE syndrome, and we have also 2 mutations that are specific for more than 50% of the DOC8 patients. So we can screen just for these 2 mutations if anyone needs to do prenatal diagnosis. In 2016, we started collaboration also with Necker for the Mendelian susceptibility of mycobacterial diseases and for CMC. 2017, we started to do collaboration with Harvard University in a large category and array of patients and for ALPS in Vienna. In 2018, we started whole exome sequencing in collaboration with Zurich for T- B+ SCID, and these data are very, very important because we thought that this category lie in the interleukin-7 receptor deficiency, and the results come on the otherwise that JAK3 is the most common in Egypt, and it accounts for 44% of this category, and 30% of the other patients are for the interleukin-2 receptor gene deficiency. In 2019, we started to do Sanger sequencing in our center for the JAK3 mutations, and we initiated interleukin-10 receptor RA and RB for very early onset IBD. Then we also sent a lot of samples by the Jeffrey model pilot study through Invitee for NGS sequencing, and it gave us a lot of results for some of the patients' phenotype, like patients with non-homologous end-joining genes, mutation, and this category wasn't well known previously. in Egypt. In 2020, we started Sanger sequencing of ITGB2 for the diagnosis of lead, and we do a lot of prenatal diagnosis for this category as those patients are miserable. They don't have any other alternative except for bone marrow transplantation if they have a living sibling. Results of this testing show that they have two mutations which are very high common for lead deficiency, and also later on a new mutation arose in half of the patients with lead gene mutation. In 2021, we started NGS sequencing for a panel of 54 genes. This allowed us to know a lot of also genes in immune dysregulation and patients with phenotype like CD4 lymphopenia and eczema, like CARD11 deficiency. Now we started a new panel of 450 genes in Egypt, and it's an ongoing project that have a lot of results that will help us later on. So in our publication, we show that in Egypt, we are following up around 1,496 inborn error of immune deficiency patients. Around 33% of these patients doesn't need to undergo the genetic diagnosis because a definitive diagnosis is reached by flow cytometry in 77% of them, and a probable diagnosis is reached depending on their clinical phenotype and other laboratory testing. Only 1,000% under genetic, 1,000 patients undergo genetic analysis. Of these 1,000 patients, around 496 were diagnosed with cystic fibrosis and familial Mediterranean fever, which is very high incidence in Egypt. And 504 patients were included in genetic testing. 44% the genetic diagnosis wasn't reached yet, and in 56% the inborn error of immunity diagnosis was reached based on Sanger sequencing in around 38% of the patient and NGS in another 36%. Whole exome was only done for 24% of the patient and FISH for 1.4%. So we started to make our own algorithm for the diagnosis of the patients. We categorize patients according to the EUS classification. And for SCID patient, we started to see if they are T-, B-, NK+. We asked ourselves if they have got microcephaly or not. If microcephaly is present, we started NGS sequencing. We are seeking for either ligase deficiency or Kernunu. If microcephaly is absent, we targeted Sanger sequencing of REG1, REG2 gene. We started with the REG2 gene, the most common hits that account for around 68% of the cases. If no mutation is seen in the REG2, we complete in the REG1 and then we do the NGS at last. For T-, B+ SCIDs, we see suggestive family history, and if the patient is female with suggestive family history, we started Sanger sequencing of interleukin-2 receptor gene. If it is a female or a patient who has undergone interleukin-2 receptor, which is normal, we started Sanger sequencing of JAK3, and after doing these two genes, only 27% of the patient in this category need to be done for NGS or whole exome. For T-, B-, NK- SCID patients, we started ADA by flow cytometry. If it is defective, so we still search for the mutation by Sanger sequencing, and if not defective by flow cytometry, we started the NGS from the start. For combined immune deficiency, less profound than SCID, we have a lot of patients with DOC8 deficiency. In this category, around 56% of the patients have DOC8. This is diagnosed by flow cytometry. We only do sequencing if the patient wants to make a prenatal diagnosis. Other categories are done by NGS and whole exome. For combined immune deficiency with syndromic features, if the patient has autoimmune manifestation and neurological impairment with hypocalcemia, we started Sanger sequencing of the PMP. If a mutation is found, that's fine. If not, we started to perform NGS and whole exome. Dardor syndrome is diagnosed by FISH, so we doesn't need any other testing. And any other category like STAT3, interleukin-6, or ORI are done by NGS and whole exome. For patients with predominant antibody deficiency, we have started now to do the Bruton tyrosine kinase by flow cytometry, and the results are very good. If the patient wants to do prenatal diagnosis, we can do it by Sanger sequencing. Other categories are done by NGS and whole exome. For immune dysregulation, LRBA account for about 54% of our Egyptian patients. If it is defective and we need to make a diagnostic by molecular, we do NGS. If not, we started Sanger sequencing of CTLA4, and then we go up for the next level of NGS and whole exome. For a very early onset IBD, we make Sanger sequencing of interleukin-10 receptor RA and RB. And in the defect of the phagocytes, patients with neutropenia undergo next generation sequencing. Patients with lead are diagnosed by Sanger sequencing, and as I said before, it gives a lot of information, and we have only 3 hits that are very common in Egypt. Cystic fibrosis patients are diagnosed by reversed hybridization, and CGD patient, we have very good data in flow cytometry by DHR, and then categorization of the patient depending on the net pH by flow cytometry. If the patient has SIBA gene, we are targeting the exon 5 hot mutation. Then we complete the rest of the exon. For NCF1, we do exon 2 screening for the hot mutation, and then we perform the Sanger sequencing of the rest of exon if the patient doesn't fit in this mutation. For CYBB and NCF2, we do Sanger sequencing for all the exon, as all mutations are variable in our patients. For autoinflammatory disorders, which are very common in Egypt, you have 483 with FMF. We do reverse hybridization and give a lot of beneficial results. For diseases of the innate immunity, like CMC, we make Sanger sequencing for STAT1. If a mutation is found, okay, and then we go to the next level by NGS and whole exome, and other categories are done by NGS and whole exome. Also, the genetic diagnosis have helped us to identify the pathogenic variant, and these allow to offer genetic counseling services for a lot of families. And as you see now, a lot of categories have been done for the family service and genetic counseling, including RAG1, RAG2, lead, ADA, and MHC class 2 deficiency. And we are doing it for a lot of families. And as you see in family 1, they have gone prenatal diagnosis in 4 pregnancies. So to summarize, in some cases, the molecular testing may reveal a genetic diagnosis that was not expected by the patient phenotype. So it's very important to see that, and sometimes we have got associations. We sent whole exome for T- B plus KID, and it showed a homozygous mutation in the RAG1 gene, and another patient had RAG2 gene mutation. Also, patient with autoimmune lymphoproliferative syndrome and has got 20% of T cell receptor alpha beta cell, and we were diagnosing it as ALPS. And whole exome sequencing revealed homozygous pathogenic variant in interleukin-12 receptor beta 1. Studying the candidate genes in each disease phenotype will help to find a founder pathogenic variant or a hot exon, and also a linked gene inheritance. Some mutations are very common in specific areas and with geographic distribution, so it helps us to target this mutation. It also helped to validate the result of other easier and less expensive diagnostic testing, especially that with flow cytometry, like the Wiskott-Aldrich, LRBA, DOC8. When we started this testing by flow cytometry, we were still trying them, and we need to confirm them by another method. Especially, we sometimes have to use monoclonal antibodies that were not previously validated for flow cytometry. So when the genetic result confirms the flow cytometry, this make us use these monoclonals later on. Finally, each center should follow its own algorithm to select the patient.
Conclure, s'il vous plaît.
Okay. Each center should follow its own algorithm to select the patient that benefit most from genetic testing and the families who need the definitive diagnosis or genetic counseling. This is the references used, and this is the PID Cairo University team. I would like to thank them all for their efforts, especially Dr. Aisha Marsafi, the founder of the PID unit in Egypt, and thank you very much.